So as we said, the single-celled fungi are called yeasts, and yeast reproduces asexually by budding or sexually through spore formation. Yeasts are usually larger than bacteria, around 3 micron across. They are usually round or egg-shaped. They do not have flagella or cilia, but contain most other eukaryotic organelles. Unlike molds, yeast grow as smooth colonies on agar plates. So looking at molds now, the body or thallus of a mold consists of long branched thread-like filaments called hyphae, and growth occurs at the tip of the hyphae. Then lots of these hyphae intertwine together to form the mycelium. So most hyphae are divided into separate cells by end walls called septa, and these hyphae are called septate hyphae. And there are tiny holes in the septa which allow for the rapid flow of nutrients from cell to cell along the hyphae. This is known as cytoplasmic streaming. The hyphae in some fungi, however, such as bread molds, are not separated by septa. Instead, they are formed by large cells containing many nuclei. And this arrangement is described as a coenocytic hyphae. So coenocytic hyphae. So these coenocytic hyphae have no septa, so no crosswall. So growth occurs at the very tip of the hyphae, and this is the region where the cell wall extends to produce a long hyphal tube. And the cytoplasm within this tip is filled with numerous vesicles, and these are produced from the Golgi apparatus. And chitin synthase is actually present in some vesicles to enable the chitin production for the cell wall. And in the higher fungi, these vesicles can be detected under a microscope as a round spot with a diffuse boundary. And this body is known by its German name, the Spitzenkorper. And its position determines the direction of growth of a hyphae. So fungi are eukaryotes and have a complex cell structure. So as eukaryotes, the fungal cells contain a nucleus enclosed in a nuclear envelope where the DNA is wrapped around histone proteins. And fungi usually have a haploid genome, so that's a single set of unpaired chromosomes. Fungal cells also contain mitochondria and a complex system of internal membranes including endoplasmic reticulum and Golgi apparatus. So the cytoskeleton in the fungal cell composes of microtubules, microfilaments, and intermediate filaments. The microtubules act as a tramline system for the membrane-bound organelles, whereas the microfilaments are involved in cytoplasmic contraction. And the intermediate filaments are responsible for the cellular tensile strength. So many fungi are dimorphic, which means they can change their forms. And this occurs in response to changes in the environment. Some fungi change their form depending on whether they're causing disease or growing saprophytically. For example, animal pathogens are filamentous in the saprophytic phase and yeast-like in the pathogenic phase. One species example is the Histoplasma capsulatum, which is a dimorphic fungus that grows in soil, exposes hyphae, and when spores are inhaled, they become more like yeasts to survive in human organs and cause chronic lung disease. So reproduction in fungi can be either asexual or sexual. In fungi, sexual reproduction often occurs in response to adverse environmental conditions. 2 mating types are required. When both mating types are present in the same mycelium, it is called homothalic or self-fertilizing, whereas heterothalic species require 2 different but compatible mycelia to reproduce sexually. And mycelia is the plural to mycelium. In most fungi, the 2 haploid nuclei, one from each parent, do not fuse straight away. So more often than not, the cytoplasmic fusion occurs first, and this leads to a dikaryotic stage where you have 2 haploid nuclei coexisting in a single cell. The process of cytoplasmic fusion is called plasmogamy. This literally means marriage of cytoplasm. The next step in sexual reproduction is karyogamy, which means nuclear marriage. During this phase, the two haploid nuclei fuse together to form a diploid zygote. This diploid phase is short-lived. Meiosis takes place next in the gametangia organs, in which gametes of different mating types are generated. At this stage, spores are disseminated into the environment. So this slide shows sexual reproduction in the zygomycete life cycle. And zygomycetes are an important group of fungi, as we will discuss later on. So sexual reproduction starts when conditions become unfavourable. Two opposing mating strains, labelled plus and minus in the diagram, must be in close proximity, and then each strain produces a different hormone that causes their hyphae to form gametangia. First, the gametangia fuse. This is the process of plasmogamy, which we described previously. And then the nuclei fuse to form a diploid zygote. The zygote develops a thick, rough black coat and becomes a dormant zygospore. These then remain dormant until environmental conditions become favourable. When the zygospore germinates, it undergoes meiosis and produces haploid spores, which will then grow into a new organism. So asexual reproduction is the other form of reproduction shown by fungi, and this can be carried out in several ways. So one way is the parent cell undergoes mitosis and divides into two daughter cells by a central constriction and formation of a new cell wall. This formation of the new cell wall is called transverse fissure, and this is shown in figure A. Or mitosis can occur with budding, which happens with yeast. The formation of asexual spores often accompanies asexual reproduction and is usually used as a means of dispersal. So different types of spores can be produced. You have atherospores, which are formed when a hyphae fragment, which is shown in B. Then you have chlamydospores, which are thick-walled survival spores, which are formed under certain growth conditions. You can see these in figure C. Sporangiospores, on the other hand, develop within a sac called a sporangium at the hyphal tip, which is shown in figure D. And then you have conidiospores, which are spores that are not enclosed in a sac, but are produced at the tip or sides of the hypha, shown in figure E. And then finally you have blastospores, which are produced from a vegetative mother cell budding. So you can see that in figure F. So this slide looks at yeast production in more detail. Yeast can reproduce asexually or sexually, but asexual reproduction is more common. Yeast reproduce asexually by a process known as budding, and this vegetative growth occurs when nutrients are plentiful. Yeast can also reproduce sexually by means of sexual spores called ascospores, which result from the fusion of the nuclei from two cells followed by meiosis. Meiosis occurs in nutrient-poor conditions. So if we have a look at the life cycle of brewer's yeast, which is S. cerevisiae, when nutrients are abundant, haploid and diploid cells undergo mitosis to produce haploid and diploid daughter cells respectively. Then when nutrients are limited, diploid cells undergo meiosis to produce 4 haploid cells that remain bound within a common cell wall. Upon the addition of nutrients, 2 haploid cells of opposite mating types come into contact and fuse to create a diploid cell.